Hello and welcome to this Introduction to Microbiology video. I'm Dr. Alid Roberts, and in this video we're going to look at various aspects of the influenza virus, from its basic biology through to its ability to cause pandemic infections. And so one of the most important aspects we need to remember when we talk about influenza is that we are not talking about a specific single virus. You see, there are many different types of influenza virus out there, all of which form part of the Orthomyxoviridae family, and within this family there are 7 different genera, of which 4 are related to the influenza virus. Now, the 4 influenza genera are named the alpha, beta, gamma and delta influenza viruses, which for those of you that are good with the Greek alphabet will notice they are essentially referred to as the influenza A, B, C and D viruses. Now, there are some key similarities and differences between each genera, starting with their host organisms. So influenza A, B and C viruses are associated with human infections. However, for the most part, only influenza A infections are of concern to us. And if we look at animals, the Delta influenza virus is capable of only animal infections, not human infections. However, all other influenza viruses can infect both humans and animals. However, again, it's the influenza A virus infections that are of primary concern. Now, from this point on, we are going to focus solely on the alpha influenza virus and their associated infections, as these are the most important from a clinical perspective. And so, influenza infections are commonly associated with acute febrile respiratory infections, meaning they infect many different aspects of the upper and lower respiratory tract, causing short-term infections that result in some form of fever, which has become sort of a hallmark feature of influenza infections. Now, the incubation period is roughly between 1 and 4 days, and due to the way the virus replicates, an infected person will typically feel fine for a number of days post-infection, and then all of a sudden they'll be hit with debilitating symptoms such as fever, fatigue, aching, a dry cough and a sore throat. Now, for the most part, an acute illness normally resolves itself within a week without medical intervention. However, some members of the community are at a heightened risk of chronic infection. This is where they fail to recover and require some form of medical intervention, and in the most severe cases, they may never actually recover, and around 500,000 people die annually from influenza infections. Now, people with influenza infections are most contagious in the first 3 to 5 days after symptoms start to develop. This increases to over 10 days in children and can be even longer in people who are immunocompromised and unable to effectively clear virions from their system quickly. Now, if we move on to the epidemiology of the disease, the influenza virus is spread in a number of different ways, from aerosolized droplets coughed or sneezed up from the respiratory tract to various fomites such as door handles that have been contaminated with these aerosolized droplets. In addition to this, there are some zoonotic transmission routes, and this is where an infection will be passed on from an animal to a human. Now, some groups of people who are at a heightened risk of influenza infection include those such as children and the elderly, who might have an underdeveloped or deteriorating immune system, those with chronic conditions such as heart disease, underlying respiratory diseases such as asthma, and those with diabetes. Other groups include those who are immunocompromised or who have an altered immune system, such as those who are pregnant. And finally, we have healthcare workers. Now, this is a slightly different group, as typically they are a healthy group of people. However, they are put at an increased risk because they will perhaps find themselves surrounded by patients suffering from influenza infections on a daily basis. Therefore, the likelihood of them receiving an infective dose is heightened. Now, one final aspect of epidemiology for the influenza virus is the distribution of the disease. Infections for the disease are global, with different variants of the virus affecting different regions. In addition to this, there is usually an increase in infection rate during the winter months. Now, when we move on and look at the pathogenesis of the disease, infection occurs at the mucosal surface, usually of the upper respiratory tract, where acute infections occur. However, it can progress down into the lower respiratory tract, resulting in chronic infections. Now, once the initial infection starts, it will spread to adjoining epithelial cells and can then be spread further afield to other hosts via coughing and sneezing. And we need to remember that the spread of the disease is governed by cell tropism, which relates to the ability of a virus to infect certain cells. Not all cells contain the receptors used by the influenza virus, and therefore the influenza virus is limited to what it can infect. So moving on, we're now going to focus on the virus itself. So our influenza A virus is an enveloped virus, meaning it has a host-derived phospholipid membrane that forms its outer surface and defines the virus. Within this are what we call M1 matrix proteins. This helps to provide the virus with structural support and help our influenza virus to escape from host cells after they have been infected. Now, there are a number of proteins that are associated with the membrane, starting with hemagglutinin, of which there are around 300 to 400 of these per virus. Now, our hemagglutinin has a homotrimeric structure, meaning it is composed of 3 identical subunits made of various carbohydrates and amino acids. Now, the structure of one hemagglutinin can differ to another, and in fact there are 18 different subtypes of this protein that have been identified to date with various different properties. Now, one important aspect as an outward-facing protein is that it has antigenic properties, meaning it can be detected by the host immune system. However, its outward-facing nature is crucial to its cell-binding function during the initial stages of host cell attachment. It also helps to fuse various membranes once the virus is taken into a host cell, releasing the viral genome into the unsuspecting host. The next surface protein is neuraminidase, of which there are around 40 to 50 of these per virus. And our neuraminidase has a homotetrameric structure, meaning it is composed of 4 identical subunits made of various carbohydrates and amino acids. Now, just like our hemagglutinin, the structure of one neuraminidase can differ to another, resulting in 11 different subtypes of this protein, which again affects various properties. And again, as an outward-facing protein, it has antigenic properties. However, the function of neuraminidase lies in its catalytic head group, which allows it to enzymatically release the virus from the host cell after it has infected them. And this means neuraminidase is a crucial protein, because without it, the virus will remain attached to the host cell when it is released, making it even more difficult to infect other cells. Next, we have nuclear export proteins, which are membrane-associated proteins that are located within the virus, and so they are not outward-facing, and therefore they are not antigenic. Essentially, their role is in genome movement, helping to move the viral genome to where it needs to be within the host cell. Next, we have the M2 ion channel proteins, which is a transmembrane protein channel, meaning it is able to bridge the internal environment of the virus and the external environment. Now, they don't just freely allow the movement of materials into and out of the virus, they are what we call a pH-active channel or proton-selective channel, which means they only allow the flow of hydrogen ions or protons across the membrane. Now, the M2 ion channel is involved in multiple stages, with a key stage being the initial acidification of the virus, which helps to facilitate the release of genetic material into the host and the replication process. Now, within the structure, we have the genome, which for the influenza A virus is 13.5 kilobases split across 8 linear segments of single-stranded negative sense RNA, with 3 large, 3 medium, and 2 small segments. And encoded on these 8 strands are the 10 different proteins required to make a fully functional virion. Now, associated with each linear segment of the genome is what we call a cap structure. This is a viral RNA-dependent RNA polymerase composed of 3 proteins, PA, PB1 and PB2, and together they allow the production of viral messenger RNA as well as replicating the viral genome in the nucleus of the host cell. And finally, as a virus, we can't forget about our capsomer proteins, which when joined with the viral genome segments form nuclear proteins and ultimately the nucleocapsid of the virus. Now this virus, despite appearing spherical, has a helical nucleocapsid structure that is masked by the envelope. The main function of the nucleocapsid is for protection, but it also has some functions in transcription and the packaging of the viral genome into new virus particles that are produced within the host cell. Okay, so moving on, now that we've seen the morphology of our influenza A virus, we are going to revisit cell tropism, which as I said earlier is where the virus is only able to infect certain host cells due to the receptors present on its surface. So here you can see the surface of an epithelial cell of our upper respiratory tract, and it has a number of sialic acid receptors present. Now these sialic acid receptors have a specific tropism that allows it to recognize and bind to the influenza A virus. And that is because extending out from this receptor are chains of galactose that bind via an alpha-2,6 glycosidic bond to a molecule of sialic acid. And it's this arrangement that allows the receptor to bind to the hemagglutinin of influenza A viruses, which can then be internalized via the process of endocytosis. Now all of this is possible because of the specific receptor and proteins on the surface of the virus, acting like a lock and key mechanism. Now some cells do not possess this receptor, and some cells possess this receptor but have a slightly different structure by which the sialic acid is bound in an alpha-2,3 configuration, and this is just enough to effectively stop binding, and if we stop binding and attachment then we don't get host cell infection. And so previously when we talked about certain influenza viruses infecting humans and some others infecting animals, it can be down to this kind of cell tropism we've just discussed. Now as we mentioned previously, some viral proteins are external facing, meaning they are on the surface of the virus and therefore they have antigenic properties, meaning they can be recognized by our immune system and subsequent reinfections can be dealt with swiftly. However, many viruses, including the influenza virus, can accumulate changes in their surface antigens, and when enough changes accumulate, there will be an altered host immune response, as the host will no longer recognize the virus. Now, there are two types of antigenic variation observed in the influenza viruses. First, we have antigenic drift, which is where small changes or minor mutations accumulate in the genome over time, eventually resulting in a new variant with an altered immune response. The second mechanism for antigenic variation is antigenic shift. This is where there is a major alteration of the genome or a reassortment event that results in some form of new subtype which might not have been seen before in humans, and we can visually represent these two mechanisms on a graph. So if we plot genetic change over time, we can see that during antigenic drift there is a change in the original genome over time due to point mutations, with each mutation altering the genetic code slightly. Antigenic shift, on the other hand, is where there is a large change in the genome at a single time point due to genome reassortment. So this is where a cell might be infected with two different types of influenza virus, and different segments of the genome are packaged into different virions, creating a slightly new variant. Now this is particularly concerning when we have changes in our surface antigens such as hemagglutinin and neuraminidase proteins, as this can lead to significantly altered influenza infections. Now as I mentioned earlier, we have some 18 different hemagglutinin subtypes and 11 different neuraminidase subtypes within our influenza A family. And each of these hemagglutinin subtypes can match up with each of the neuraminidase subtypes, creating many different combinations, some of which are common amongst human infections, others against animal infections, with some being relatively rare. Now, there aren't just differences in the surface antigens of influenza. Due to the nature of the genome, diversity is seen in all aspects of the influenza virus. However, it is these antigenic changes which are key because they alter how our immune system perceive and recognize the virus, meaning changes can lead to seasonal epidemics and even pandemic variants. And for those that don't know, a pandemic is an occurrence of an infectious disease over a wide area affecting many communities within a specific time period. And for our influenza virus, pandemic influenza is the result of what we call a reassortment event, where different antigens are packaged into the virus, creating a novel subtype not seen before. And so to visualize this, here we have an influenza A virus that infects humans. Now it can do this because human epithelial cells have sialic acid receptors that have a specific alpha-2,6 configuration, and therefore human influenza viruses attach to these and cause infection. Now in addition to this, influenza A viruses can also cause animal infections, with birds being the most common. Now it can do this because bird epithelial cells have sialic acid receptors that have a specific alpha-2,3 configuration, and therefore animal influenza viruses attach to these and cause infections in animals. Now this isn't necessarily a problem. Human influenza viruses infect humans and avian influenza viruses infect birds. However, it's not as simple as that. You see, pigs have epithelial cells which have both alpha-2,3 and alpha-2,6 sialic acid receptors, meaning they can be infected with both human and avian influenza viruses at the same time. And this makes the swine a mixing bowl, where elements of the human virus could potentially mix with elements of the avian virus, creating a brand new variant which has different characteristics based on the two parental strains of the virus. And because of this, the virus might technically become less effective with reduced mortality and transmissibility, which would be the best case scenario. Alternatively though, it might become even more effective as a virus, with high mortality rates and increased transmissibility rates. And this might lead to a pandemic, as we would be immunologically unprepared, meaning our bodies have never seen such a virus, and it would therefore be more likely to spread and cause disease amongst different populations. Now, pandemic variants aside, influenza is somewhat of a self-limiting disease in that we can typically overcome it with some rest if we have a fully functional immune system. However, if this was to become compromised, we would need some form of intervention. Now, in terms of treatment interventions, options can be relatively limited, and that is because they are limited molecular targets for our influenza virus. We need to remember it is a virus with a very small genome that uses host cells to replicate, and so if treatments target these processes, then we might end up with toxic side effects on our own cells. Thankfully, we do have some treatment options, starting with oseltamivir, which is a neuraminidase inhibitor and blocks this protein from working, which essentially means the influenza virus isn't able to be released from a host cell, stopping the spread of infection to adjacent cells. Next up, we have rimantidine, which is an M2 ion channel inhibitor, and this blocks the ability of the virus to release its genetic content into the host cell, meaning the host cell never receives the instructions to produce more virions. And finally, there is baloxavir, which is an RNA-dependent RNA polymerase inhibitor capable of inhibiting the viral polymerase so that it is unable to replicate the viral genome or produce the required viral messenger RNA. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.